Welcome! Today we're going to talk about chapter 17, or at least 17.1, point 2, and point 3. There are two other sections in that chapter that I think we'll get through on Wednesday. Today there is a sheet that we will work on that corresponds to a lot of what we talked about today. Next class there will be a little bit more of a lab activity, still paper pencil, but a little less worksheety, that we'll do after we finish chapter 17. And on Friday I hope to talk about at least parts of chapter 18. So, this whole unit is about DNA. What did we talk about last time? With chapter 16.
Oh, mitosis.
I mean, like last class.
Oh.
What did we talk about?
We did the lab.
No, that was,
Chi-squared.
we did chi-squared 2 classes ago.
Oh.
Replication of DNA.
Yeah, the structure of DNA and replication of DNA. That's right. Thank you. Thank you for remembering. Today we're going to talk about how DNA functions. And we're going to dive into some pretty deep detail, especially in the reading, a little less so up here. So, to this point, when we've talked, say, in last unit about genetics, we've just kind of said like, oh, you have a gene for something, so you have a trait. Well, there's all this stuff that goes on in between the gene and the trait. And that's chapter 17 and chapter 18 to some extent. So this chapter starts out, they give this example of normal pigmentation and albinism in these donkeys. And essentially what's happening, donkeys have donkey DNA, and like any other living thing, there are stretches of DNA, and segments of your DNA are called genes. And those genes are going to consist of a sequence of bases, which we represent with what 4 letters? Say a letter!
G.
G, C, A, and T. Good. Those are your DNA bases that create some sort of sequence that we represent as a letter sequence. Now here's the deal. Just because you have a gene, well, when you have a gene, it does lead to a certain phenotype. But in between the gene and the phenotype, you have 2 big processes, one called transcription and one called translation, that lead eventually to the trait being expressed. So you have a gene, which is a segment of DNA. That gene is going to basically write a message in the form of a single-stranded molecule called messenger RNA, mRNA. We've talked a little bit about RNA before, a little bit, but here it's finally kind of showing up. This mRNA message is going to go to ribosomes, which are the organelles that do what? Make protein. A ribosome will only make protein, though, if it has a message to read. So when a ribosome reads this message and makes a protein, that's what we call translation. And that protein then, in this case, is an enzyme that drives forward a reaction that produces a brown pigment that makes a normal donkey color. If there is a mutation, like with albinism, then it doesn't work that same way. The message will be different wherever the DNA has been changed. And if the message is different, the protein is different. And if the protein is different enough that it doesn't help make the pigment that this donkey normally makes, then the donkey won't make the pigment. So it's not just gene to trait, it's gene to mRNA going to a ribosome, which makes a protein, and that protein will somehow help express the trait. Okay, so that's the main point here in the beginning, is that a gene isn't just a magical spell that makes you a certain way. A lot of the beginning of this chapter goes into kind of the background and scientific history of how we made some of these discoveries. A lot of our understanding of how a gene is eventually expressed as a trait started with experiments on bread mold. So they would grow mold in these plates and give them certain nutritional substances to grow on called growth media. And they used X-rays to damage the DNA of the mold. It would create mutants. Some of these mutants would not be able to grow unless given a certain nutrient. So in this case, what they found is they could create mutant molds that would not survive. They would not grow unless you gave them this amino acid called arginine. And they were eventually able to show that for these cells, for this mold to make this arginine that it needed to grow, it was actually 3 different enzymes that drove 3 different series of reactions that allowed the cells to make this arginine. And these mutants were not making one of these enzymes correctly, and so they weren't making arginine, so they couldn't live unless you gave it to them because they couldn't make it themselves. So, bread mold, who knew? But that's what the first section of chapter 17 goes into a bit, is some of these experiments where we were able to damage DNA, find that that resulted in the inability for those mutants with this damaged DNA, they were no longer able to make a certain protein, and thus they could not express a certain trait, like living without needing this amino acid being given to you. Okay. So to get back to, and this is more the second and third sections of the chapter, to get back to what exactly is going on, I wanted to show you this eukaryotic cell here to give you an idea of where some of this is happening. They do have this eukaryotic versus prokaryotic cell here, this bacterial cell without a nucleus. That's what a prokaryote is for the most part. I'm not too worried. We talk about prokaryotes a lot in this chapter, but I'm not too worried about this. In this case, with the eukaryotes, the DNA is stored in the nucleus. The DNA is going to guide the production of mRNA, which is not just one quick step, and we're going to talk about that. And then the mRNA is what will leave the nucleus and find a ribosome, and that is where you start to make a protein, which, remember, proteins start as what we call polypeptides, many amino acids being stuck together. The ribosome can only do that if it's got a message. The making of the mRNA, again, is transcription. Using the mRNA and making a protein with it is called translation. Now, in prokaryotes, where there's no nucleus, nothing leaves the nucleus because there isn't a nucleus. So It's a little bit of a quicker, more efficient process because there's no nucleus, boundary that needs to be passed. Okay. A little bit more detail. So we're kind of zooming in a little bit, or just looking at it from different perspectives here. Double helix shape on the DNA molecule. At different points along the molecule, you can find different genes. So when we say gene, we just mean a segment of DNA. Notice we have the two backbones. It's double-stranded. Everywhere there's an A, across from it there's a T. And C's and G's are also what we call complementary to each other. Remember, there's a 3 and 5 prime end. The other strand runs anti-parallel. So this is the 5 on the other strand and the 3 on the other end. One of these sequences will be read. Okay? And that is based on a variety of factors. In this case, in this diagram, they're showing what we call the template strand. The template strand is the strand that gets read. Now, I don't want any misconceptions about it always reads the top one or anything. That's just how this diagram looks. Either this sequence or this complementary sequence will be the sequence that gets read, and the message will come from that. In this case, we are just being told that it's this ACC, AAA, CC, GA, GT, that this top sequence here, in this case, happens to be the one that's being read by the cell. Well, when the cell reads this message, it will transcribe a message, and you make a sequence of RNA that's, again, called mRNA. Now, we talked about the differences between DNA and RNA a long time ago. The D versus the R is the result of the sugar that you have. Remember, the backbone is a phosphate sugar base, that's the structure of it. Deoxyribose is the name of the sugar in DNA. Ribose is the sugar that you find in the RNA structure, so that's one difference between them. RNA tends to be a single strand versus double-stranded. And there is a letter, or a base, that we see in RNA sequences that is not in DNA sequences. And what is it? U. RNAs don't have T's, they have U's instead. I have no idea why. It's just evolutionarily, that's how the chemicals developed and these processes developed. But if you look at this template strand that we are using, so look at this top one, ACC, the message that you transcribe from that is UGG. A writes a U as a message, and the C's come out and the message is G's. They're complementary, they match. AAA, UUU. CCG, GGC. A writes out in the message a U, the G a C, and then the T in DNA corresponds with the A. A and T always go together, but anywhere you would have a T in RNA, the letter U or the base uracil is used. So this is our message that we wrote from this template strand. Now, we don't really get into this in the reading that much, I don't think, for next class, but I wanted to go ahead and talk to you about how this message is read in general, and then we're going to go back to transcription and go into a little more detail. Every 3 letters of the message is known as a codon. This is a word you need to know. A codon is a 3-letter message, and each of these 3-letter messages, each codon, is going to tell a ribosome which amino acid shows up at that point. So if you notice, this codon here codes for this amino acid. This codon codes for a different amino acid, and so forth, as you read along. We have cracked this code. We've known this code for a long time, and this chart, as scary as it may look if you've never seen it before, is a chart that I will have on the test that you will need to use for even the worksheet today. And it's not that hard to read the chart, even though it does probably look a little overwhelming if you've never seen this before. But this is an mRNA chart, and what it allows us to do is translate a codon into a specific amino acid. So if we go back to our first codon here, like UGG, remember that? UGG. UGG. The first letter is U. So these are all the codons that start with U. The second letter was what? G. So these are all the UGs right here. And then, I mean, if you really need to, you can go to this side of the chart and find the G, but there it is. There's UGG. Once you get those first 2 letters, you're in the right box. That is for tryptophan, which we rarely write out the full name of the amino acid. We use these little 3-letter abbreviations. And by the way, I never use these, but there are actually single capital letter abbreviations for a lot of these. And some of them make sense, like alanine being A and valine being V. And then you get tryptophan, well, they use W to represent that. But I never use those. TRP is good enough. That is an amino acid abbreviation, and it's a different amino acid from the other ones. Now, if you notice, some amino acids have multiple different codons that all code for it. Leucine is an amino acid that has these 2 codons, all 4 of these, and I think that's it. But that's 6 different codons, and if any of those 6 show up in the message, leucine is the amino acid that is used there, abbreviated LAU. Are there any weird codons on this chart? Huh? Any weird ones?
Yes.
Yes? What are they?
And AUG.
Yes! There are some that are called stop codons. If a ribosome ever reads a stop codon, it stops adding amino acids to the chain and translation is over for that mRNA message. So stop codons stop the translation process. And those are UAA, UAG, and UGA. Which you can remember that because UGA can't stop Florida. Now,
Or Alabama.
LOL. This is a codon for an amino acid called methionine, but it's also called a start codon. Ribosomes will not start adding amino acids together to make a protein unless they read AUG. That AUG, Ningambam, is a start codon. AUG is a start codon. And he will tell the ribosome when to start amino acid production or translation and the production of a protein. Keep that in mind. That's going to come in on this worksheet because you might have letters leading up to a start codon. None of them are read or translated. You have to have that start codon first, and then the process begins. This chart, which you have all these slides on your coursework page, this chart will be used for different activities that we do. You're going to have a long string of letters that represents an mRNA, and I'm going to ask you to show what that protein would look like, or at least what that amino acid sequence would look like. You're going to have to come back to this. It's in your book, and you have it as a slide on the chart. We don't talk much about this year, but before we move on and dive into the actual details of transcription and translation, I did want to show these two pictures. You can take genes out of a genome of one organism and put them into the genome of another. And if done correctly and all the important parts of the gene are there, that organism might start expressing that new gene. We have taken the gene for the enzyme luciferase, which is an enzyme that makes light-emitting chemicals. Fireflies, or lightning bugs, whatever you call them, use that enzyme to glow, to make their little flashes. Well, scientists, 40 years ago, were able to take that gene out, put it into the genome of plants, and you can grow plants that have this eerie yellow glow because they make the firefly gene. It's a plant expressing the gene of an insect.
Didn't they do that with cats one time?
They have done it with cats now, yeah. They even have I don't think they used the firefly gene. I think they did more of this, which is a phosphorescent jellyfish gene that they used. Yes.
Did they cross? And that's not genetic,
There's really no limit to what genes you could cross from one species to another. And some of this stuff we really benefit from. We can take the human gene for insulin, put it in bacteria, and just grow plates of bacteria or tubes of bacteria that just excrete insulin, harvest it, and that's how we pharmaceutically manufacture insulin. We used to have to go to slaughterhouses and take all the pig pancreases and process them and not squeeze out literally, but squeeze out the pig insulin, and that's what diabetics had to use. This is, pretty life-changing. Question? Or did you have a question?
I was gonna say, since I was a little kid, there was a thing back home, there was a liger, a cross between a tiger and a lion.
Yes. Now, there are, that's not really genetic engineering, but you can cross what we would call almost different species. You can have Cherkees, which are chickens and turkey breeds, the ligers, tigers and lions.
Who's naming all these things?
Zedonks, which are zebra donkey.
Sure.
And in fact, there is a set of nomenclature rules for naming these things, and it actually matters in some cases whether the mother is one species or the father's the other. You can have a Zedonk or I don't know how else you would do.
Donkzy?
Donkbra. And you actually, like, depending on which one the father or mother was, I don't know if it's the mother or father, but their species name is kind of in front. It's not technical, but it is something we do.
The Zedon.
Yes.
Have they done anything like that with like humans? Are they like?
It is based on like worldwide agreement that human genetic tampering is
Kind of sick.
basically globally illegal. Now, I'm sure there are some countries that aren't really part of the global community that are doing who knows what.
It'd be pretty cool if you
Or,
glow in
like
the dark.
cross human with plants.
Yes, there are probably really fascinating possibilities that you could get. I think the problem is it's just ethically so touchy and sensitive. There's too many things that could go wrong, and basically you just, to create some bizarre version of human life that we don't know what effects it would have. But we do probably do a lot of primate research. We're very good about going, well, it's not technically human, it's just a chimpanzee. So I'm sure, we make our advances in those areas. Anyway, getting back real quick to transcription. Yes, I would not recommend having the phosphorescent jellyfish gene inserted into your child so your baby will glow. The problem is that it sounds simple on the surface, like, oh, I would just make a protein that made me glow and I'd glow in the dark. How cool would that be? But that protein might be very similar to, say, insulin in its structure. And you might glow in the dark, and it might cause you to starve because you are not processing your sugar correctly. And that's horrifying, so it's you don't know what the consequences could be sometimes. So that's why we tend not to do this on humans, and it's a lot of bacterial and plant things we do. So yeah, it can get out of control. I'll say that. All right, so what we're looking at here, from top to bottom, is the original DNA strand. All right? Now, there is an enzyme that is actually making the messenger RNA. That enzyme is called RNA polymerase. ACE ending tells you it's an enzyme. The rest is RNA polymer. Well, polymer just means many parts. So this is an enzyme that makes a stretch of RNA, one part at a time. So it's got this, like, on this diagram, it looks kind of like a guitar pick or something. So this guitar pick right here is this RNA polymerase enzyme that attaches to the DNA. Where does it attach? Well, we're going to get into this a little bit more on the next slide. But it's at a promoter region of your DNA. Every gene is going to have a sequence called a promoter. And it's actually not the start codon. It's even before that, which we'll see in a second. But we'll actually talk about it on the next slide. So there is a region on each gene called a promoter, which is kind of the starting attaching site for this RNA polymerase. The RNA polymerase then runs along the DNA strand that it's reading, and it has to actually break the two strands apart temporarily so it can read one of the strands. And as it's, you can skip down to like kind of this third step here, as it's running in this direction, and in this case it's reading this bottom strand, so as it's running from the 3 to 5 direction, it's producing the mRNA. That's what you see here in red. So it's reading the DNA as it goes along, and it's kind of pooping out the RNA message. It's just kind of producing it and letting it, trail behind it. Once it gets to a certain signal at the end, the RNA polymerase detaches, and you have what's called, they're calling it here an RNA transcript, which is your mRNA. But sometimes this needs to be cleaned up a little bit. We're going to talk about it. But that's basically transcription. The RNA polymerase attaches to the DNA strand that it's going to read, separates it from the other strand, runs along it, reading the DNA, and producing the message based on what that DNA says. And then we have our mRNA here at the end. Now, again, it doesn't start at the start codon. That's more in translation. That's what the ribosome is looking for in the message. The RNA polymerase, and don't worry about the Roman numerals. I'm not going to worry about the different versions of RNA polymerase that they have. The RNA polymerase, like I said, is going to start or attach to the DNA at this place called a promoter. And specifically, it's looking for one of my favorite terms in biology. It's looking for a Tata box.
Okay.
Now, I know it's a weird that we made words out of the A's and the T's here, but the Tata box is a region of DNA where you only have what?
The Tata.
The T's and A's in the sequence. Now, I guess they could have called it an AT-AT box, but AT-AT, I think, is one of the vehicles in Star Wars.
True.
So, it got the name Tata box because there's a lot of times a lot of tatas or A's and T's. This is where it's looking, this is what it's looking for and where it's going to attach. Now, your book goes into a lot of detail about what we call transcription factors. These are just proteins that kind of are around and also attach and help the RNA polymerase kind of get into position. But it's very important that you have this enzyme start at the right place, because that's where it's going to start reading, and based on what it reads, it's going to write a message. And that message is going to be used to make proteins that are very complex, so you want the message to be correct. So there is a way, and it's basically called the promoter, for a cell to get this polymerase in the right spot. And then again, in red here, this is the beginnings of the mRNA, the beginnings of the message that it writes. Now, look, because there's a reason I'm intentionally talking about the Tata box, even though it's not terrible, it doesn't seem terribly important, but there's a fascinating angle to it. This enzyme has to separate these two strands so that it can read the strand it needs to read and create a message. Well, if you have to separate the strands, do you remember what I said is actually holding the two strands together? What's between the A's and the T's and the C's and the G's?
Hydrogen.
Hydrogen bonds. Weak intermolecular forces exist between the A's and the T's and the C's and the G's. You have to break the hydrogen bonds that hold the two strands together in order to separate them. Now, there are two letters that between them will have two hydrogen bonds, And then there are two letters that between them have 3 hydrogen bonds. What's easier to break, 2 bonds or 3 bonds? 2 bonds. 2 bonds are easier to break apart. Okay? It's simple math. Which 2 letters form only 2 bonds between them?
AG.
A and
Two.
T.
Oh, the Tata.
A Tata box is the easiest place to separate the strands, which is what the polymerase enzyme needs to do. So it's kind of this beautiful, it's the perfect starting spot because it's the easiest spot to start. Isn't that neat? Okay, so notice they keep calling it RNA transcript. They don't call it mRNA yet. Here's what happens, and this gets weird. A lot of your DNA is junk. It's old viruses that predecessors in your family tree 20 million years ago got and ended up living with and passing on. That codes for pieces of small virus that don't even exist anymore. There are all kinds of copying mistakes that your predecessors have given to you. I think they said 1% of your DNA is actually genes that you use.
We should just get rid of the rest.
And 8% of it is actually instructions for viruses that viruses injected into your ancient predecessors millions of years in the past. You're actually, more of your DNA codes for virus parts than even codes for you.
We should just get rid of it.
And I would not recommend trying to cut out the junk DNA from your genome. You'd have to do that to every cell of your body to be successful. So, there is a lot of cleaning up that needs to be done a lot of times. So here is, and again, I know some of this stuff looks crazy and complicated. This is what we call pre-mRNA. It is the transcript. It is just the raw message that the RNA polymerase kicks out during transcription. It's made up of what we call exons, which I wish they had the word up here. These dark red sections are called exons, and the lighter kind of pink regions are called introns. There's also a cap on one end and what we call a tail on the other end. It's called a poly-A tail because it just goes A. There's a bunch of just A's all in sequence at the end. Now, these caps and these tails help the message actually get out of the nucleus and to thread themselves into the RNA, into the ribosome, and tell the ribosome when the message is over. So they have a function. The introns are actually what we call non-coding kind of segments. There are enzymes that cut out parts of the message that your ribosomes don't need. And then they glue together all the exons, which are called that because they express themselves later in translation. And so you have to cut out I don't want to say the useless pieces, some of these introns can be used for something, but they cut out the important parts and glue it all together. And it's not until other enzymes, and I'm not going to go into all the details here, but it's not until you clean up and remove the most important parts and then glue it all back together do you have what we call the mRNA molecule. A cap, the part that codes for something that you need to build, and then a tail. And last slide, we will get into translation more next time, but basically once you take out your introns, you have these different exons left. And a lot of times, these exon, the important parts, the express parts of your message, the mRNA message, they're going to code for different parts of a protein. Remember a long time ago when I talked to you about primary, secondary, tertiary, and quaternary structure?
Yeah.
Yeah, so in this message, you have like these secondary structure polypeptides, and then they're glued together to make a larger tertiary structure. And these different parts of this protein are going to come from typically different exons of the message. Just to give you an idea of how this process plays out. Now, this is the last slide in the sequence, but I want to quickly just point this out to you. This, in a eukaryote, is happening all within the nucleus, where your DNA is. And this is kind of your detailed look at transcription. Okay? So this is a detailed look at transcription. We did kind of blow up kind of the beginning here and talk about where it all starts, where the enzyme actually bonds to with the promoter region. And this is also transcription. So until you get, or when I say transcription, I'm really talking about production of the messenger RNA. Now, next class, I'm going to show you how you go from the mRNA message to an actual chain of amino acids at the ribosome. And we're going to go into that kind of detail for that process. But we're going to stop there. And what you're going to do is you're going to, and this is on your coursework page, but I have paper copies for you guys here. You're going to try this sheet here that I'm going to grade, but it's not due today. But I wanted to give you an opportunity for some more grades that aren't, giant test grades. So I'm going to put a few of them right here. Do your own. You can work with someone, but I want everyone to have their own sheet, and I can help you with it. And if you're online, you can do that. Could you hit stop the video?
Yes.
It's